Welcome everybody. This is our fourth and last lecture regarding carbohydrate metabolism. We will discuss glycogen metabolism, and our objectives are to be able to describe the structure of glycogen as a storage polysaccharide, and to explain the role of glycogen in energy homeostasis, to compare and contrast liver and muscle as glycogen storage sites, to list the reactions of glycogen with synthesis and breakdown, to describe the regulatory mechanisms of glycogen metabolism, and to discuss the role of UDP glucose as an activated glucose donor, and finally to correlate glycogen storage diseases with the respective steps in the pathway. At any time, if you draw a blood sample and estimate it as a source of glucose, it will be a combination of diet, gluconeogenesis, and glycogenesis, depending on the last meal, when was the last meal, and the state of the body. So glycogenolysis is an important source of blood glucose. Glycogen itself, as you remember, is a branched polymer of alpha-D glucose. It is a homopolymer since it is constituent units are all alpha-D glucose, and it is a hexosome because when it is hydrolyzed, it will release only hexoses. And it functions as a major storage form of carbohydrate in animals, corresponding to starch, of course, in plants. And the main function of glycogen, aside from being a storage storage form of glucose, is to act as a readily available source of hexose units whenever they are needed. The main storage sites are the liver and the muscle. The liver can store up to 6% of its weight as glycogen, while muscles, particularly skeletal muscles, can store up to 1% of its weight as glycogen. So which one comprises the largest store of glycogen? It is the muscles because it has a greater mass, and the greater mass corresponds to much more glycogen compared to the liver. In fact, muscles can store up to 3 to 4 times as much glycogen that is stored in the liver. In a 70 kilogram a normal adult in a post-absorptive state, the liver can contain up to 72 grams, while the muscles will contain up to 245 grams of glycogen. Which one is selfish and which one is selfless? Muscle glycogen acts as a readily available source, however, for the use within the muscle itself, while the glycogen of the liver is used for maintaining the blood glucose level. So it will be released into the circulation to reach whichever organ or tissue or cell that needs it. The liver is selfless. It avails its glycogen for whichever needs it, while muscle glycogen is reserved for the muscle use only. When we discuss glycogen metabolism, we will go through glycogenesis, which is glycogen biosynthesis, and glycogenolysis, which is glycogen breakdown, and finally we will discuss some related disorders. Glycogenesis occurs mainly, as we said, in muscle and liver because they are the main storage sites, and glycogenesis involves a special nucleotide of glucose, an active form of glucose, and that is uridine 5-diphosphate glucose. So this is our uracil, this is ribose, that is diphosphate, and this is our glucose. This whole molecule is an active form of glucose, UDP glucose. UDP glucose is a critical activated intermediate in carbohydrate metabolism in general, and particularly in the synthesis of glycogen, as we will see today in glucuronic acid pathway and in synthesis of lactose, as we have seen in the last lecture, as well as in the synthesis of complex molecules such as glycoproteins and glycolipids. Glycogenesis requires the action of 5 enzymes: hexokinase glucokinase, that is similar to the first enzyme in glycolysis, a phosphoglucosidase, a UDP glucose pyrophosphorylase, glycogen synthase, and amylo 1,4 1,6 transglucosidase. The first reaction is phosphorylation of glucose to glucose 6-phosphate, similar to glycolysis. And this is catalyzed by hexokinase in muscle and glucokinase in the liver. ATP is used in an irreversible reaction. The second reaction, glucose 6-phosphate, is converted to glucose 1-phosphate by phosphoglucosidase. And as you can see, this reaction is reversible. In the third reaction, the glucose 1-phosphate is converted to the active form of glucose, the uridine diphosphate glucose, UDP glucose, by the action of UDP glucose pyrophosphorylase. UTP, uridine triphosphate, is used as a source of energy, and there is subsequent hydrolysis of the pyrophosphate by a pyrophosphatase, ensuring that the reaction goes in the right direction irreversibly. The fourth reaction is catalyzed by glycogen synthase, in which carbon 1 of the UDP glucose forms a 1,4 glycosidic bond with the fourth carbon of a terminal glucose residue of glycogen. UDP will be released from the UDP glucose. However, it cannot start from scratch. Glycogen synthase cannot just join three glucose molecule with another glucose. It has to act in what we call a glycogen primer. This is a condition for the action of glycogen synthase. So what is a glycogen primer? A glycogen primer consists of glycogenine plus some residues of linked glucose molecules. Glycogenine will be elongated by the action of glycogen synthase, adding glucose molecule sequentially in a 1,4 glycosidic bonds to form glycogen as we know it. The glycogenine is a 37 kilodalton protein which has an enzymatic activity, particularly a glucosyl transferase activity. It becomes phosphorylated at a certain tyrosine residue. So this is glycogenine. It is a protein that has an enzymatic activity. It can add up glucose molecules. Particularly, they will be linked to a certain tyrosine residue within the glycogen. So the first glucose molecule will be linked to the protein. What do you think this bond or this link is here that connects the first glucose molecule to the glycogenine protein core? Let me know your answer. And then up to 8 molecules of glucose will be added. Now this structure that is formed of glycogenine plus 8 molecules of glucose is called a glycogen primer. On top of this, glycogen synthase now can add up the further glucose molecules. Glucose molecules. In this short video here, illustrates the 3-dimensional structure of glycogenine, and this is a whole molecule of glycogen. This glycogenine or the glycogen primer in the center of the molecule. And this glucose molecule, this glycogen molecule is most likely from a muscle glycogen. because in muscle glycogen, the ratio of glycogen molecules to the glycogen primer or to the glycogen will be 1 to 1. In the core of each glycogen molecule will be one glycogen primer and one glycogen. While in muscle, for each glycogen, there may be more than one glycogen molecule. The last reaction is branching, and as we know, glycogen is a branch molecule. The branching or the branching enzyme is called amylo 1,4,1,6 transglucosidase because it transfers a minimum of 6 residues to a next chain to form the branching bonds at the 1 to 6 glycosidic bonds. There is a condition also here for a branching enzyme to act. The length of the branch must be at least 11 glucose molecules in length for it to be able to cut 6 residues and transfer them to a next branch. So this is part of the glycogen molecule. Glycogen synthesis can extend it each branch by adding glucose molecules. The newly added glucose molecule are in black. Each branch now contains this one, I think it is 11, this one around 12 or 13. The branching enzyme can now come into action. The branching enzyme cuts at least 6 residues and transfers them to a neighboring branch, creating alpha 1 to 6 glycosidic bond. Now this end here is free for adding more glucose. This end here is available for adding new glucose molecules by glycogen synthesis as well as this branch. And then each one of them, when it is 11 or more, will be subjected to the action of the branching enzyme. Now, our glycogen molecule is complete. We need to degrade it or to release some glucose to avail it, for example, for the RBCs in need in between meals. Glycogenolysis, the breakdown of glycogen, is not the simple reversal of glycogenesis. In fact, it is a separate pathway catalyzed by different set of enzymes. For glycogenolysis, we need also the action of 5 enzymes, including a glycogen phosphorylase, alpha 1 to 4 glucan transferase, amylo 1 to 6 glucosidase, a phosphoglucosidase, and finally a glucose 6 phosphatase, similar to that in glucogenesis. A question for you here is regarding the enzyme phosphoglucosidase. How many metabolic pathways does it participate in, at least in carbohydrate metabolism? Write down your answer. The first reaction in glycogenolysis is phosphorylated breakdown, whereby our alpha 1 to 4 glycosidic bonds are broken down using inorganic phosphate, and glucose 1-phosphate will be released as a product. Phosphorylated breakdown is catalyzed by glycogen phosphorylase, which acts on the outermost glucose residue, and it starts the cutting from the very end and coming closer or proximal to the branching point. It will not reach it. It will continue cutting as long as it is 4 residues away from the branching point. And the coenzyme for this glycogen phosphorylase is vitamin B6 in the form of pyridoxal phosphate. So pyrophosphorylase will start cutting, for example, from this side. It will cut this one, and it will stop because it is now 4 molecules away from a branching point. And this is what we mean by phosphorylated breakdown of 1 to 4 glycosidic bond with release of glucose 1-phosphate. The second reaction of glycogenolysis is transfer of a trisaccharide. We said the glycogen phosphorylase has stopped acting at 4 residues away from the branching point. From these 4 residues, a trisaccharide, that is 3 connected glucose units will be transferred from one branch to another by alpha-1 to 4 glucan transferase, and this will expose the branching point. So this is our 4 residues that were left from the action of the debranching from the previous reaction of glycogen phosphorylase. This outermost 3 residues will be transferred by glucan transferase to expose the branching point. Now the branching point is accessible for the third enzyme, as a debranching enzyme, which catalyzes hydrolytic splitting of the bond 1 to 6 glycosidic bond, and it is called amylo 1 to 6 glucosidase. And now we don't have a branch, it is a usual, or it is just a length of glycogen residues. It will be subjected to the action of glycogen phosphorylase. Glycogen phosphorylase will act upon the length of the branch until it reaches a branching point where the confined action of amylo 1 to 6 glucosidase and transferred by the glucan transferase will ensure that the branching point is accessible to continue the action. The fourth reaction is reformation of glucose 6-phosphate. We said the glycogen phosphorylase releases glucose units as glucose 1-phosphate. The phosphoglucosamidase interconverts the glucose 1-phosphate back to glucose 6-phosphate. And glucose 6-phosphate, of course, will be subjected to the action of glucose 6-phosphatase to release glucose. Please note that glucose 6-phosphatase is found in the liver and kidney, which are the sites for glucogenesis, but it's not present in muscle. So muscle cannot release glucose in the blood. Even if glycogenolysis took place in muscle, all the released glucose from glycogenolysis will be trapped within the muscle cells. It will not be released into the blood. Why? Because muscle doesn't have the last 5th enzyme, the phosphatase, and that is why the muscle cannot avail its glucose to other organs through the circulation. It is not merely selfless. It just doesn't have the action of the glucose 6-phosphatase. How glycogenesis and glycogenolysis are regulated? They are reciprocally regulated through controlling glycogen phosphorylase and glycogen synthase, and the mechanisms involved are chemistry and covalent modification and hormone regulation. Glycogen synthase is active in the dephosphorylated form and inactive in the phosphorylated form. This is in contradiction to glycogen phosphorylase, which is active in the phosphorylated form and inactive in the dephosphorylated form. Let's see how does that take place. Regarding allosteric regulation, glucose 6-phosphate is an allosteric regulator, active allosteric effector on glycogen synthesis. So when there is a lot of glucose, it will be phosphorylated by glucokinase and hexokinase, and this glucose 6-phosphate will activate glycogen synthase and activate glycogenesis. Why? To store all the large amounts of glucose as glycogen. At the same time, glucose 6-phosphate is a negative allosteric regulator of phosphorylase, particularly in muscle. So when there is a lot of glucose 6-phosphate or glucose, this will inhibit or inactivate muscle phosphorylase. It will tell the muscle to stop glycogenolysis. There is no need to break down its glycogen because glucose 6-phosphate is abundant. AMP, which signifies low energy stores, activates muscle phosphorylase to release glycogen, of course, for the use by muscle, while glucose, which signifies abundance of energy, turns off or inactivate the phosphorylase of the liver. It tells the liver to stop glycogenolysis because there is a lot of glucose. And as for the covalent and hormonal regulation, cyclic AMP-dependent kinase will phosphorylate and inactivate glycogen synthase while it activates phosphorylase, so it will activate or stimulate glycogenesis and reciprocally inhibit glycogenolysis. Calcium-dependent kinases also does the same thing. It will inhibit glycogenesis and activate glycogenolysis. A protein phosphatase removes a phosphate group and then thus it will activate the glycogen synthase and stimulate glycogenesis, and reciprocally it will dephosphorylate and inactivate the glycogen phosphorylase and eventually inhibit glycogenolysis. Insulin, as a hormone, is produced in response to high blood glucose concentration. So there is a lot of glucose. This glucose needs to enter the cell and to be used as an energy source through glycolysis, and the extra glucose has to be stored as glycogen. So when there is a lot of glucose, there is a lot of, or the insulin will be released and it will activate the glycogen synthase to store all that glucose as glycogen, and at the same time it will inhibit the phosphorylase. It will stop glycogen breakdown because there is no need for it as long as the glucose is abundant. Now we come to the disorders of glycogen metabolism, mainly a group of disorders that are called glycogen storage diseases. They are genetic disorders, so they are inherited in an autosomal recessive pattern. They are characterized by deposition of abnormal type or quantity of glycogen in the tissue. So the quantity could be normal, but the type is abnormal, or the type is normal, but there is a lot of glycogen more than the normal stores in the tissue, and this will disrupt, of course, action and the function of the cells. How does glycogen storage diseases manifest? They will manifest as hypoglycemia because this glycogen will not be available as a readily source for glucose. So glucose will be low in the blood. Lactic acidosis because glycolysis will proceed through the anaerobic pathway, and ketosis because the ketogenesis will be activated to try to provide extra sources of energy. Heart failure because, for example, the accumulation of glycogen itself and the inavailability of energy. Liver failure as well. Diminished exercise tolerance because of the accumulation of glucose in addition to inavailability of energy for the muscle contraction. Hemolytic anemia because, as we know, RBCs are dependent first and foremost on glucose as an energy source. This glucose will not be available from glycogenolysis because glycogen is abnormal, whether in type or in quantity. How are the conditions managed? Depending on the specific type, frequent meals to replenish glucose in the circulation, limited exercise, and to replace the deficient enzyme, for example, pyrocomplement enzymes, and liver transplantation is also an option. This is a table that illustrates the types. They are written in Roman letters and their names and their respective deficiency. Glycogen storage type 1 or Jenks disease is related to glucose 6-phosphatase deficiency, while type 2 POMS disease is caused by deficiency of alpha-4 glucosidase. Type 3 has many clinical forms: POMS disease, Graves' disease, or limbic stenosis due to the deficiency of the deep branching enzyme alpha-1 6 glucosidase. The fourth, Glycogen Storage Disease or Anderson disease or amyloidectonosis is due to deficiency of the branching enzyme, the alpha-1 6 transglucosidase. Type 5, McArdle syndrome, is due to deficient muscle phosphorylase, while deficient liver phosphorylase will lead to type 6, which is called heart disease. The seventh type, Stargardt disease, is due to deficiency of phosphofructokinase, and the eighth type is due to the deficiency of the liver phosphorylase kinase. A question for you is regarding the seventh type, Stargardt disease, due to deficiency of phosphofructokinase. Why is the deficiency of the glycolytic enzyme phosphofructokinase is classified as a glycogen storage disease? This concludes our talk. Thank you very much.